Thousandth of a milliliter?
1 millimeter, 1 milliliter.
Oh, 1 milliliter. Oh,
Did I say millimeters?
milliliter.
Milliliter. 1 milliliter, okay? So P1000 is 1 milliliter. How we're going to use these again is we set them up so that the digits, we don't go over, we don't go out of their range. So if I'm wanting to pipette something that's 20 microliters, which pipette do I use?
P20.
P20, okay? Use the right tips. This one uses the blue tips, the other ones use the yellow or the red tips. Today we're going to do pipetting. Our goal for the first lab is to just pick up some skills, okay? We're going to be using a lot of pipetting and a lot of microscopes over the course of the next 6 months. So we want to kind of gain some of those skills. So the first thing we're going to do is we're going to get out the microscopes, and we have a kind of a puzzle we're going to put together. It's a dilution puzzle. So we have, this is called diatomaceous earth or diatoms. Does anyone know what that is? Or use it in the garden?
I've seen it for all kinds of weird things on TikTok. So,
Probably.
like
What?
skincare or like
Ah,
gut health or
sure.
something. Yeah.
It kills bugs. It's really good at killing bugs because diatoms are little photosynthetic algae type things, but they are made out of they're like almost petri dishes, one smaller than the other, but they're glass. And so when they break apart, they're glass spicules, and you can kind of see all the glass, and it's really kind of neat. The glass is really fine, so it destroys insects and worms and stuff like that, but for you, it doesn't really affect your skin or anything like that because your skin's too tough. So it can exfoliate, things like that. So what we're going to start with is 100 milligrams of diatomaceous earth into a beaker. So how many milligrams are in a gram?
1,000.
1,000. So 100 milligrams is 0.1 grams. Okay, so 100 milligrams of diatomaceous earth and 3 mils of water. You're going to take the bulk pipette right here, these guys, and you're going to take a drop, put it on a glass slide, look under the microscope, and you're going to see how many diatoms there are. The hint, there's going to be trillions. Okay? Your job is to do dilutions so that you end up with 1. Okay? So dilutions are really hard. So if I start with a volume this big, okay, so this is a decent amount of water, and I want to do a dilution, 1,000-fold dilution. How much water would be needed? You'd fill this room full of water. Or, what if you took 10 microliters of water out and put 1,000 microliters of new water in? How much of a dilution is that? Taking 10 out, put in a new beaker, then add in 1,000 microliters. That's 100-fold dilution. So you can see where you can use actually a very small amount of water to do a huge dilution. So if I want to do 1,000-fold dilution, I can take 1 microliter out and put 1,000 microliters in. There's your 1,000-fold dilution. So if you start with 1 trillion and you do 1,000-fold dilution, how many do you have?
1 million.
1 billion.
1 billion.
Then you do another 1,000-fold, you have
1 million.
1 million. Then you do another 1,000-fold, you have
1,000.
1,000. Another million-fold, you have 1. Right? So there's how you do your dilutions. So you shouldn't use more than 4 milliliters of water. You don't need to fill the room. You don't need to go to a swimming pool to dilute this. You only need a small amount. So that's our goal. I don't really care about what you actually see. I care that you're learning how to use dilutions again, using your pipette, and the microscopes. How many of you use microscopes before? Okay. The biggest thing with microscopes is to go slow. You don't need to run the stages or anything into each other, okay? So that's the biggest thing. Just go slow and easy. Use your coarse, use your fine, get it into a field of view. If you have a question, ask. Microscopes are one of these things where it literally will take you 1,000 hours to get good. It takes a while. So we just want to get some experience with it. So we're going to look at this. You're going to look at your diatoms. You're going to do your dilutions. Hopefully this doesn't take more than a half hour. And then I'd like you to look at 4 or 5 different things under the microscope, okay? We have algae up here, somewhere, right there. Pull a hair. Look at hair. If you're wearing mascara, pull an eyelash. You want to see worms or insects or things like that. You might be able to find mites. I had a student earlier today. She found egg masses and mites on her eyelashes because she was wearing mascara. Pretty common. Lovely, but you have things like that in you. Pull, cuticle, skin, whatever you want to look at something different under the microscope. Again, I do not care what you're looking at. I want you to get used to the microscopes and the pipetting, because that's what we're doing 2 or 3 weeks a year. Then we're going to move on to the other lab. Okay? All right, how do we feel? Good. Let's do the worksheet pretty quickly. Grab out a microscope, and then in your group, we're going to probably work in pairs-ish. Grab a beaker, come up with, you got the weight paper here, we're going to put that in. Get 100 milligrams of diatomaceous earth, start doing your dilutions, and life is good.